Good morning, and I must say thanks to the organizers to invite me for this presentation. We are actually working on some 3-4 different vaccine projects at Hillman Laboratories, and I would like to take you through one of our projects. I would first like to mention a bit about Hillman Laboratories because many of you may not be aware of the details. So we were instituted in 2009 and based at New Delhi, and this institute is actually funded by Merck and Wellcome Trust as a joint venture, and we started our lab operations in 2011. And this is basically in memory of the legendary vaccinologist called Dr. Maurice Hilleman. Now let me come to the topic, and the topic is a serious topic, looking at the seriousness of the other diseases also. And the agent is actually, it affects any age, it kills many, either it is infant, kids, adults, and it doesn't even spare the survivors. The 2 kids, so this particular kid got amputated for the limbs, and you could see this, could see the little one. And of course we need to keep on moving on, even the life is difficult. So I hope I have made you very much serious about the disease which I am going to talk about, and the culprit is not the vaccine, it is a Diplococcus, and for 2 of them they may not be important, maybe we need more. I don't want you to be too loose, so I would like to take you directly to the topic, that is Neisseria meningitidis. The contents of my presentation would be the need of an affordable multivalent meningococcal conjugate vaccine, challenges and avenues in developing the vaccines, promises from the synthetic vaccine technologies, and what we do in this area in our laboratory, and of course I'll conclude that. So what is the need of an affordable multivalent meningococcal vaccine? So I would first talk a little bit about meningitis. It is an inflammation of the meninges of the brain as well as the spinal cord. And the causes of meningitis can be bacterial, viral, or sometimes fungal, and mostly I would talk about the bacterial meningitis, which is actually in form of meningitis or septicemia, and sometimes combined. Septicemia is much more dangerous than meningitis because it leads to the further complications. The bacterial causes, there are several, but Neisseria meningitidis is one of the major which I am going to talk about today. And the symptoms are quite a few, the fever, vomiting, rashes, I showed you some slides, and headache, delirium, drowsiness, pains in the muscles and joints. So let us talk about some epidemics or the spread of the disease in the majority of African regions. So before, obviously I'll be talking more of the recent epidemics. In 1976, there was transmission of the meninge A serogroup disease from Africa to Asia, and Africa happens to be the hub of the meningitis cases. So 1996, 20,000 deaths reported, and of course the suspected cases were in lakhs. In 2000 and 2001, several pilgrims who were attending to Hajj, they were infected with meningitis W because the meningitis W is very much prevalent there. And in 2002, Burkina Faso, the meningitis W, it struck 13,000 people in the same country and killing 1,500 people. In 2007, epidemics in Burkina Faso again, 22,000 people, suspected cases, 1,500 deaths approximately. 2009, 14 countries where the active surveillance has started from 2004 onward, 88,000 cases and 5,300 deaths. And last year itself, the data is, which is the minimum deaths after 2004, since the active surveillance has started, 19 African countries, 11,000 cases, 12,000 cases, and around 1,200 deaths. And the bug, in case of the epidemics, may cause up to 20% mortality, and of course I have already shown the serious disability that can happen after even survival. And this is the very famous meningitis belt in sub-Saharan Africa, and there are almost 21 countries where it happens in epidemics happen in frequency of 4 to 10 years. But this is not all. So meningococci, they are available globally, and they can cause disease, but the prevalence of the major serogroups are like this. So in Africa you can find all the 5-6 serogroups which are most prevalent out of the 13, but in other continents obviously the seroprevalence vary. And this is how it changes. It is not a fixed seroprevalence. In Europe and in USA, it is mostly C and Y. I am not talking about B here because this is out of my context, because B cannot be prevented by the conjugate vaccine. So I am just going to focus on A, C, Y, W, and X serogroups. So these 6 serogroups, they cause the majority of the diseases out of the 13 serogroups known. So C and Y, in case of Northern America, if you go to Southern America, then W also comes up in the extreme south part of the South America. Europe, it was mostly C earlier, but after the introduction of menin C vaccine, there has been a significant reduction, and now B only remains, and this should be covered by the Bexsero, that is the new vaccine that has been licensed in England at least. Let us talk about Middle East. Then A, C, and W in case of the Middle East. And if you see the percentage of W cases, they are very significant here, and there is a good amount of transmission from the Saudi Arabia to the other countries because of the Hajj population, and that's why there is a mandate that whoever is going for Hajj, they have to get vaccinated now with ACYW conjugate vaccine or polysaccharide vaccine. Asian countries, obviously again the country-wise, and even in New Zealand and Australia, the variation is there for the serogroups. But if we talk about meningitis belt, where the maximum deaths happen, it was 78% for meningitis A cases, which has reduced to 10% approximately in 2013 because of the introduction of the massive campaign for MENA vaccine. However, if we see that there is a shift from MENA to now MEN-W, and even the MEN-X case is 2.5% to 11%, so MEN-X is also on rise. And I'll be showing some latest figure just now. If we talk about England, earlier it was mostly C and B. C was reduced because of the C vaccine introduction. B is going to be tackled, but if we see the latest reports, 2015 February, we can see that the W cases are rising. And recently, actually, the ACYW vaccine has been launched to replace the MEN-C vaccine, which was part of the vaccine immunization program there. And this is a very recent letter from the director Path, that there has been an epidemic, very recent epidemic in Niger because of meningococcal serogroup C, and that's how he is recommending that there is a specific need for the multivalent vaccine, so you cannot fix a particular serogroup to a particular region. So the answer is the meningococcal conjugate vaccine, not monovalent, it should be multivalent. And what are these conjugate vaccines? If some of you are not aware of, this is the polysaccharide, capsular polysaccharide originating from these bugs. And this is a T-independent antigen, and it cannot work in case of kids below 2 years of age. And then there is conjugated to a protein, which is a carrier protein, through a linker, and this conjugate vaccine gives you a T-dependent response. But these multivalent vaccines presently available, there are 3 of them: Menectra, Menveo, and Nimenrix. Novartis developed Menveo, and now because of taking over by GSK, it now is being held by GSK, and GSK's Nimenrix has now, obviously there are takeovers going on. So the cost of these vaccines are not small, and this can never be afforded by the poor countries. So that's why there is a very big need of the cost-effective vaccines, and we need to provide right serogroup combinations. If at all we have to go lesser than 5, and there is a need to reduce dependency on few of the vaccine manufacturers who can produce low-cost supplies, and the existing low-cost conjugate vaccines can be the cost can be reduced by adopting new technologies. So I'll be talking a little bit about that, but before that I would like to show you just forecast about the demand of Men-A vaccine, and now it can be reflected in terms of the multivalent vaccine also. So if we go till 2034, so this is actually the 2014 data. There is a big campaign going on with Benefrivac. So the campaign will reduce because it is supported by Gavi, and as the countries are getting graduated, so there will be less and less support from the Gavi to these countries, and more requirement for the cost-effective vaccine will be there. So what are the challenges in developing these conjugate vaccines? First is that there are 13 serogroups and there are regional variations, and the earlier slide I showed, that was the major serogroup, but if we see, this is a whole lot of variety of infections we can expect all around the globe, and the serogroup keep on changing based on the age groups as well as the different time intervals. This is US data. If we see 10-year interval, there is a big difference between the serogroup prevalence, and even in all age groups versus the infants. So if we see specifically men C is quite less here, and infants less than 1 year. So the combination of the serogroups is very important. There are very complex processes to develop these conjugate vaccines. You have to first produce the polysaccharide, which should meet the specification, and if it is 5 different components, it has to be, it is a very costly affair. You activate the polysaccharides, produce the carrier protein by different process, and then activate this. Both of them, they are conjugated to come to a bulk conjugate stage. And then you produce lyophilized or non-lyophilized liquid formulation. And there is a significant analytics required, especially for the multivalent vaccines. I must say that around 50 or more analytical tests, they are required to be released for the release of a multivalent vaccine from start till finish. So it is a lot of work, and it is very difficult to achieve a cost effectiveness in this case. So what are the possibilities to develop a cost effective vaccine? So there are novel technology solutions which we can go for. So we are actually targeting several of them at our lab, and these are novel improved polysaccharide production processes in the conventional vaccines. Antigen synthesis, which is a very novel approach and which I will be highlighting in my presentation. Reverse engineering, say for example meninge B vaccine. Novel and improved conjugation processes, several different conjugation processes can be tried. Novel formulations like thermostable formulations. Novel analytics like multiplexing immunoassays. So let us come to the topic of synthetic vaccines. in today's world, there are several things that are being used, so which are chemically synthesized, and many of them they are organically synthesized. How the interest in the synthetic vaccine started for the conjugate vaccine, it was the effect of chain length that was studied for the group B streptococci, and the scientists found out that a very small length was also able to give rise to the appropriate immunogenicity. And because the polysaccharide structure has been elucidated very well nowadays, and there has been a significant improvement in the organic synthesis in the chemistry world, and that's how there was an interest. And in 2004, this was the first success with the first semi-synthetic conjugate vaccine developed for Haemophilus influenzae type B vaccine in Cuba. And this has not stopped there. There has been some of these recent articles I could mention. One of them is obviously from our own lab, and obviously the research is going on. But why there is no second semi-synthetic vaccine yet, even in the clinical trials? So there are very big challenges before you can adopt to a novel technology. So the manufacturers and regulators will ask for several questions. And these questions will be comparison of the conventional and novel technology in terms of GMP requirements, duration of production, cost of production, batch-to-batch consistency, impurity profile, analytics, stability, immunogenicity, ease of modifying structure, conjugation yield, and loss of epitopes in the conjugation vaccines. So we are actually working on answering all these questions before we can go to the manufacturers for the clinical trial collaborations. So this is a case for ACWXY, that is a pentavalent meninge vaccine, which we are trying from the conventional platform versus the semi-synthetic platform. Why I'm calling semi-synthetic is we are organically synthesizing the oligosaccharides and conjugated it to a natural carrier protein, that is tetanus toxoid or some other carrier protein. And we will also opt for some defined regional combinations if possible. Otherwise, a pentavalent vaccine will be a big advantage based on the cost effectiveness. So I like to answer a few of these questions through my these following slides. The polysaccharide production, if we see the process, there is a cell banking. You have to get the cell bank from some lab. You have to keep on paying royalty for that. Fermentation, so this is handling pathogenic organisms, and you know that what is the impact of disease. So even accident happens, then the GMP requirements are very high. PS production, purification by several steps, and size reduction because the natural size cannot be used directly for the conjugation. However, in case of the synthetic oligosaccharides, we start with a commercially available chemicals, and then we make initiation unit which can be easily linked to a linker without impacting on the natural the structure of the oligosaccharide. We make a propagation unit, and this is the case for meninge C tetramer. This is a sialic acid unit, and we combine these two to make a dimer, trimer, and tetramer. So this is actually a linear synthesis, and the option is also for block synthesis. You could combine two of these with two of these to make a tetramer and octamer. And I would say that this is an API kind of manufacturing process, and you can get this polysaccharide, oligosaccharide produced at a very low cost as compared to these. And we have been able to successfully synthesize meninge C tetramer versus octamer, meninge W, Y, and X tetramers already, along with the same linker. And I'll tell you the benefits of this linker in future slides. Because these oligosaccharides are very small, 1.5 approximately kilodaltons. That's how they can be very easily characterized as compared to the conventional polysaccharide is very difficult because they are too large and complex. Advantage in terms of size distribution, there is a limit to the size distribution in the natural polysaccharides. If we see the 3 batches of the meningococcal C polysaccharide developed in our lab, so they look, if average molecular weight, if we talk about, this is very good, same. But if we see the polydispersity, it is very big, and if we see the 3 lots of the meningococcal C, tetramer or octamer, so they will be very much fixed because it is an organically synthesized product, very defined product. Let's come to the next step of the conjugation after the polysaccharide production. So in this case, of course, the polysaccharide has to be degraded to a small length. There is no need to degrade here. In the conjugation, whatever chemistry we use, we are actually impacting on the epitopic structures in case of the natural polysaccharides. For example, I take a reductive amination chemistry. In case of serotype 19F or Streptococcus pneumoniae, the ring opens here in this case, and you are modifying this epitope. If we take another chemistry, that is the cyanylation chemistry, then also we are reducing this modification, but still we are modifying the epitopes here. And same thing will happen for reductive amination in case of Haemophilus influenzae. So you are actually modifying to a certain extent the epitopes. However, in case of the organic synthesis approach, the protein is modified by maleimide labeling, and the oligosaccharide is just thiolated on the end of the linker, so there is no impact on the oligosaccharide, and this is directly linked to the protein through the thiolation. So this is a thiol combination, and the same chemistry can be utilized for all the oligosaccharide, whatever serogroup it may be, and that is not really possible for the other chemistries. The process of conjugation is quite consistent, repeatable with high yields, and if we see the contrast of the free oligosaccharide versus the conjugated oligosaccharide and free natural polysaccharide versus conjugated polysaccharide, the benefit here is the getting the free polysaccharide out of the conjugate, which is again a big requirement from the regulatory. It will be very easy because the 2 peaks are quite separate. Here it will be very difficult to get to the limits, so you are getting a loss of the conjugate. The process is quite robust. You get very good consistent ratios. Free oligosaccharide is very much less, in most of the cases less than 3. We have made 8 batches, and the yield is approximately 35% as compared to around 20% we get in case of natural polysaccharides. The second last question we had asked is the comparability with the licensed vaccine. So we have taken a licensed vaccine, that is Menectra in this case, and compared our Men-C octamer TT conjugates with, so this is the licensed vaccine, and these bars are the octamer TT conjugates. We tested them for immunogenicity in the mice model, and we tested sera after 2 and 3 doses. So this is the post 3 dose data. IgG titers, total IgG, as well as the functional antibody titer, which is mandatory for the meningococcal vaccines, and we see that there is a very good comparability. I won't say it is superior at this time, because still there is a gap that Menectra is a tetravalent and our vaccine is a monovalent at this time. So we'll be continuing this research. We have done some research in comparison of the chain length, that is tetramer versus octamer, and we see that both of them, they give almost equivalent responses, either it is IgG or SB titers. But still we would like to take octamer as a lead candidate. There are other benefits, like we get easier ratios of the polysaccharide to protein. We have just initiated the Menex tetramer TT, which is again for the first time synthesized, and we are getting very good titers because there is no licensed vaccine available. So we have compared it with the negative control, and we get 10 to 15 fold higher titers as far as total IgGs, and we'll be waiting for the SB data. So we are working on this stability studies for these conjugates versus this conventional conjugates. And the last question in front of us was obviously the time taken for the synthesis. So it is a significant difference in the time taken. But there are solutions for that, which we have not been able to do it till now, but will be opting for as we progress further. So there are automated organic synthesis tools available, there are labs set up, and then there are of course publication. This complex synthesis could have taken 9 months to synthesize from these reactants to go to this stage, but the manuscript tells that it can be completed within 2 to 3 days by automated synthesis if we have the automated synthesizer. So there is a good possibility that we could reduce the time for the synthesis also. So let me now compile my presentation. GMP requirements, I would say that the minus is a negative attribute and plus is a positive attribute to a technology. So now I am comparing the conventional technology versus the synthetic oligosaccharide. So GMP requirement wise, because it is API manufacturing and the cost of manufacturing can be very less, so I would say, and we are not handling any pathogenic organisms, so it is quite good here. Duration of production, it is much better here as on today, but I would say that significantly longer time percentage, but scope for automation is always there, and we'll be able to answer that over a period of time. Cost of production, I would say that still we are under evaluation, but I can assume that there will be a significant cost reduction because of this. And this is not only for the synthesis, because we are getting higher yield of conjugate, so more number of doses can be prepared from the same amount of the oligosaccharide. That is also one more advantage. Batch to batch consistency is much more with the synthetic oligosaccharide, because live organisms they will not produce as per our own wish. So organic synthesis is a much more specific thing. About the impurities, protein, nucleic acid impurities, we do not expect from the organic synthesis. Endotoxin contents, we don't expect. Residuals, yes, this is because we are using so many chemicals, so these are still under evaluation how many residuals we can get, and we'll be doing this. Ease of modifying structure is very easy with the synthetic oligosaccharide, not at all possible with the natural polysaccharide. Loss of epitopes during conjugation, I have already mentioned that this is very much less with the synthetic oligosaccharides. Conjugation yield is much better because you have got a defined chemistry, same chemistry for all different conjugates. Analytics, because of the oligosaccharides being very specific. So it is very good and very robust, and regulators would actually like this defined oligosaccharide. Immunogenicity, we have seen comparability as on today, and we'll keep on exploring that. And stability also is under exploration, but I can say that the larger polysaccharide will be more prone to degradation because of the fragility, and the smaller oligosaccharide, they will have better stability. So in the conclusions, I would say among all other novel technologies, synthetic approaches to develop new conjugate vaccine is an emerging subject. Initial successes also in our lab, they have paved the way for exploration of synthetic platform technology for other vaccines. Advantages of synthetic approach over the conventional approach have upper hand, and the approach may lead to successful candidate in future. I would acknowledge MSD and Wellcome Trust. Obviously, they are the founders for our organization, and our CEO, Dr. Gill, and Dr. Jim Israel, who is already present here, and all our members in the conjugate vaccine program, our consultants, and the CROs. And thanks a lot for your patient listening to the presentation.
Thank you very much for a very interesting talk. When do you foresee that these vaccines can be commercially available on the market?
I would say that it may take at least 4 to 6 years. The reason is like this, that we have got one vaccine already is licensed by Cubans, but now the complexity has started increasing, more number of serogroups. So we have got some initial data. And then we would like to partner with the manufacturers, because we are a R&D organization. So that's what I wanted to tell, that we are open for good collaborations with academic as well as manufacturing organizations to take some of these developments further. And I think after that the clinical trials will happen, and that's how we believe that at least 5-6 years will be required.
Thank you. Any more questions?
Yeah, just wondering why you use tetramer and octamer? Why two?
Good question. Actually, the minimum what we found out from the literature was a dimer for a pneumococcal polysaccharide that was fine with the immunogenicity. But if we take the Hib example, we have actually done some other research also which I have not presented on Hib. So minimum tetramer is actually essential for the immunogenicity in case of mouse model. But if you talk about translating the mouse data to the human data, so it may not be essential that the tetramer will be sufficient. So what we are trying to make sure that from the beginning that we don't land into a problem tomorrow. So we are exploring the higher lengths also. So that's how the tetramer and octamer comes into picture.
Increasing the length of the carbohydrate can increase immunogenicity.
Yes, it can, but our MENSI data, it shows that in the mouse model at least they are not showing any difference. So basically that's size of the oligosaccharide you require for the immunogenicity, because finally it is getting degraded, the natural polysaccharide also into the macrophages or the antigen presenting cells. So we need a very small length for the immunogenicity to be generated. So that's what we are exploring, and that will be cleared in the clinical trials also.
Any more questions? Okay, then thank you very much.
Thank you very much.